Crohn's disease by Kate Hollingsworth and Michael Alday. Nurse 231, Physiology and Pathophysiology. Red Deer Polytechnic, Denise Childs, March 30th, 2023. Crohn's disease is a chronic inflammatory bowel disease alongside irritable bowel syndrome. Crohn's is characterized by its periods of remission. Crohn's can affect any point from the mouth to the anus, most commonly affects the distal small intestine and proximal large colon. So the four tunics. From the esophagus to the colon, the GI tract wall has four basic tunics. The mucosa is the innermost layer or mucous membrane, which is a most epithelial membrane that lines the lumen of the alimentary canal from mouth to anus. The main functions of the mucosa are to secrete mucus digestive enzymes and hormones, absorb the end products of digestion into the blood, and protect against infectious disease. The typical digestive mucosa consists of three sublayers: a lining epithelium rich in mucus-secreting cells, a lamina propria, which contains capillaries that nourish the epithelium and absorb digested nutrients. It contains isolated lymphoid tissues, part of the MALT, which stands for mucosa-associated lymphoid tissue, which help defend against bacteria and pathogens which freely have access to the digestive tract. Externally, the muscularis mucosae can enhance absorption and secretion. The submucosa external to the mucosa contains a rich supply of blood and lymphatic vessels, enabling the stomach to recoil. The muscularis externa is responsible for segmentation and peristalsis. The outermost layer is the serosa. The small intestine is the major digestive organ where the digestion is completed and nearly all absorption occurs. The small intestine is a convoluted tube which extends from the pyloric sphincter of the stomach to the ileocecal valve where it connects to the large intestine. There are three subdivisions of the small intestine: the duodenum, the jejunum, and the ileum. Modifications for absorption. It has adapted to absorb nutrients and has modifications including circular fold, villi, and microvilli. Circular folds are deep permanent folds of the mucosa and submucosa. They force chyme to slowly spiral through the lumen, allowing adequate time for full absorption of nutrients. Villi are finger-like projections of the mucosa. Each villus has a dense capillary bed in the core called a lacteal, which absorbs digested foodstuffs into the capillary blood and lacteal through the enterocytes. Microvilli are long, densely packed cytoplasmic extensions of the absorptive cells of the mucosa that form the brush border. The brush border contains brush border enzymes, which complete the digestion of carbohydrates and proteins within the small intestine. Histology. Between the villi, the small intestine is studded with intestinal crypts. Within the crypts, 5 major villi cells are found. Enterocytes are absorptive cells which bear the primary responsibility of absorbing nutrients and electrolytes within the villi. They secrete intestinal juice, which is a watery mixture that contains mucus and acts as a carrier fluid to absorb nutrients from chyme. Goblet cells are mucus-secreting cells found in the epithelial of the villi and crypts. Paneth cells fortify the small intestine's defenses by releasing antimicrobial agents. These secretions destroy certain bacteria and help to determine which bacteria colonize the lumen. Enteroendocrine cells, source of enterogastrones. Stem cells constantly divide and their daughter cells differentiate. Small intestine motility. 2 motility patterns in the small intestine. After a meal, segmentation is a principal form of motility. Segmentation ensures that chyme is thoroughly mixed with bile and pancreatic and intestinal juices. Also ensures that the absorbable products of digestion come in contact with the mucosa for absorption. Between meals, the motor function is primarily a form of peristalsis called the migrating motor complex. This motor function sweeps debris toward the large intestine. The duodenum contains pacemaker cells, which depolarize more frequently than the ileum, therefore moving intestinal content toward the ileocecal valve.
The major function of the large intestine is to absorb most of the remaining water from indigestible food residues, temporarily store residues, and then eliminate waste as feces from the body. The gross anatomy of the large intestine includes 10 E. coli, which is a longitudinal muscle layer of the muscularis. It is mostly reduced to 3 bands. Their tone puckers the wall of the large intestine into pocket-like sacs called haustra. Epiploic appendages are small fat-filled pouches of visceral peritoneum which hang from the surface of the large intestine. The colon has several distinct regions, including the ascending colon, transverse colon, descending colon, and sigmoid colon. Then lastly, into the anal canal. The microscopic anatomy includes thicker mucosa and abundant crypts are deeper. The crypts contain a tremendous amount of goblet cells producing mucus which eases fecal passage and protects the intestinal wall from irritation by acids and gases. Bacterial microbiota. The large intestine contains thousands of different types of bacteria. Our gut bacteria help us by recovering energy from otherwise indigestible foods and synthesizing some vitamins. Absorption. Digestion is the catabolic process that breaks macromolecules into monomers, which are small enough for absorption. Enzymes carry out hydrolysis, breaking down these molecules, and only these molecules are small enough to be absorbed into the small intestine. Absorption is the process of moving substances from inside the lumen of the gut into the body. Tight junctions ensure that molecules must pass through the epithelial cell rather than between them. The specialized structures of the small intestine play a large role in absorption. The brush border enzymes of the small intestine aid in the breakdown of these macromolecules, allowing them to become small enough for absorption. The villi allow for these monomers to be absorbed into the capillary blood and further transported within the blood. Pathophysiology. Typically occurring in the terminal ileum, the gastrointestinal epithelial barrier of the GI tract wall normally separates gut content from the underlying immune system, preventing the body from reacting to dietary antigens and resident bacteria of the gut. A crack or defect in the epithelial barrier allows for these pathogens to go through the wall and therefore increasing the chance that the immune system overreacts to known pathogenic antigens of the gut content. It is also possible that a virus or bacteria such as Mycobacterium, Listeria species, or paratuberculosis may also trigger the immune response, as the pathogens are now able to enter the GI tract via the faulty epithelial barrier, causing a response as they are foreign antigens. These pathogens stimulate an immune response due to their antigen presentation. The inflammation beginning in the intestinal submucosa due to the immune response eventually crosses the intestinal wall into the mucosa and serosa. Type 1 helper T cells initiate the immune response by activating leukocytes and cytokines, which cause further damage to the intestinal wall. Inflammatory cells such as prostaglandins and proteases are released by leukocytes, continuing the inflammatory response. It is believed that one step within the immune response is dysfunctional, causing an unregulated inflammatory response, which eventually leads to the destruction of healthy tissues. The reason for the dysfunctionality of the process is possibly due to a gene mutation or error in gene expression. Nucleotides are typically read in groups of 2 or 3, but by adding or subtracting one nucleotide, this essentially shifts all nucleotides, therefore coding for an entirely different amino acid, causing a dysfunctional protein. Damage from Crohn's extends through the entire depth of the intestinal wall. As more cells are being destroyed, the foreign pathogens move into the deep mucosa and form into granulomas, which are masses of immune cells attempting to encapsulate what they think is foreign. Eventually, because of this cell destruction and inflammation, apoptoid lesions, which are shallow ulcers, result and destroy the intestinal crypts. The mucosal lesions, such as crypt inflammation and abscesses, which progress to ulcers, eventually form into deep longitudinal and transverse ulcers, which result in a cobblestone appearance of the bowel. Within the large intestine, harmful bacteria are normally kept in check in 2 ways. Initially, the beneficial bacteria outcompete and outnumber the harmful bacteria, actively suppressing them. Second, the gut epithelium is meant to prevent bacteria from entering the body. Dendritic cells sample the microbial antigens in the lumen, then migrate to nearby lymphoid follicles within the gut mucosa, which is the malt. This then triggers an IgA antibody response restricted to gut lumen. This is supposed to prevent the bacteria from straying into deep tissues of the mucosa, which could cause a more widespread response. During Crohn's, this coexistence of the bacterial microbiota within our immune system fails, which results in the bacteria migrating into deep tissues of the mucosa and eliciting the same effect as above, activating an immune response and damaging the intestinal wall. Symptoms. The small intestine's main role is absorption, and as the intestinal wall becomes damaged, the cells, structures, and cells aiding in the absorption of nutrients are also destroyed, therefore decreasing absorption of nutrients as seen in figure 1.6. Alongside the destruction of these specialized cells and structures, the surface area is also being decreased, in turn decreasing the amount of nutrients which can be absorbed. Malabsorption, because of the damaged intestinal wall and structures, including the microvilli, villi, and brush border enzymes, then lead to nutritional deficiencies, weight loss, fatigue, and malnutrition, as nutrients normally absorbed within the intestine are now unable to be absorbed. Blood in the stool is prevalent because of the damaged intestinal wall. If damage is more prevalent in the large intestine, diarrhea will be present as the cells within the large intestine lose their ability to absorb water as they become more damaged, which causes more water to be excreted into the stool as the water cannot be absorbed, therefore causing diarrhea as shown in figure 1.5.
Treatment. Even though there are no known cures, there have been treatments to relieve the symptoms and reducing the inflammation. Glucocorticoids. They work by blocking the T cell activation in the inflammatory response, which then stops the release of cytokines and other inflammatory responses. Looking more closely, the steroid, specifically the glucocorticoids, binds to any target body cells. The binding occurs when the glucocorticoid binds to a receptor on the outside of the cell. The GC receptor is activated and carries the GC into the nucleus. The GC inhibits the transcription of activator protein 1 and factor kappa B, which are two potent transcription factors for inflammatory responses, such as cytokines. By inhibiting these transcription proteins, this will inhibit the pro-inflammatory response due to the T cells not being activated and starting the cell-to-cell communication. Another form are antibiotics. Certain antibiotics target the specific proteins that are made by the immune system. In our gut, we have a barrier of epithelial tissues that separate the inner and outer intestinal walls. The inner walls of the intestine house the normal bacteria found in the gut, and sometimes a foreign bacteria penetrates through the inner wall and causes an immune response. Antibiotics will help lessen the pathogenesis of the bacteria by inhibiting, such as inflammation, discomfort, and diarrhea. Antidiarrheals. A symptom of Crohn's disease is diarrhea, and that can be discomforting and in some severe cases lead to dehydration. An antidiarrheal is loperamide, which is a capsule, a tablet, or liquid. Loperamide works by reducing the water in the intestines and thus slows down the bowel movement. Loperamide binds to the opiate receptors in the gut wall. At the same time, loperamide inhibits the release of acetylcholine and prostaglandins, which are part of the inflammatory response. By doing so, the propulsive peristalsis is slowed down, meaning slowing down the amount of time for intestinal travel. The slowing down of intestinal travel means that diarrhea will be inhibited or at least slowed down. Loperamide also increases the tone of the anal sphincter, thereby increasing the contractions of the sphincter, and increases control and lowers incontinence. Current research. For patients who have Crohn's disease and use immunosuppressive drugs, it is an obstacle for those who have Crohn's disease and do not have the desired outcomes using the medications such as explained. A new research involves the stem cells that are extracted from the umbilical cord or the bone marrow, conducted with a mixture of regular immunosuppressive drugs, in hopes of good outcomes. There are two possible cells that have been used. There are two possible cells that could have been used: hematopoietic stem cells, HSCs, or mesenchymal stem cells, or MSCs. Using MSC cells that have been approved by the European guidelines, these were injected intravenously. The idea was that the MSC cells cause lymphocyte apoptosis, or cell death, which stopped the immune response of the body in the gut. Although sadly, the study did not yield positive results, as the MSC cells did not target their proper cells. The researchers noted that the stem cells did more adverse effects, and that further research was needed to fully conclude their findings.